So hello and welcome everyone. Thank you for joining us today for this webinar dedicated for a cutting-edge and increasingly strategic technology, supercritical carbon dioxide. So today we will have a specific focus on its use in supercritical fluid chromatography, or SFC, for the pharmaceutical industry. My name is Jean-Christian Hartemann. I'm in charge of the business development of SFP process here in India, based in India, with our trust and great partner Sanitec Engineers Private Limited, represented by Nilesh Benani, and I'll be guiding you through this session. So during these 45 minutes, we will explore the scientific, technical, and industrial potential of carbon dioxide in its supercritical state for the purification in Indian pharmaceuticals. This webinar is organized with the support of the EU-India Chambers, and before starting, I would like to warmly thank you, the Chambers, and shortly introduce them. So this organization was established in 1992 in Mumbai under the aegis of the EU ambassador and act like an independent platform uniting bilateral chambers to foster EU-India trade, investment, and technology exchange. Guided by a great team and expert committees, the Chamber serves as a key forum for business networking, collaboration, and knowledge sharing. Many thanks for their support, communication, and for those who are unfamiliar with the institution, I highly recommend you to contact the services for more information. So let us start. And for this session, I am joined with Jérémy Lagru, founder and CEO of SFE Process. So Jérémy is a French expert pioneer with over 15 years of experience in the field of supercritical fluids. In 2015, he founded his company, SFE Process, of which I'm part, in his garage. After exponential growth, the company has become a globally recognized expert in the field of supercritical solution and equipment. Today, the French SME employs around 50 persons with a global presence around the world, with an extent network of partners and representatives. SFE Process has supplied solutions in more than 40 countries across the world, with various applications for cosmetic to pharma, and is developing since several years new application of supercritical carbon dioxide for the plastic industry, aerogel, micronization, encapsulation, medical devices sterilization, and a lot of new perspective application. Today, we are also joined with Nilesh Benani, Mr. Nilesh Benani. He's the founder and director of Nielsen Nisotech System Private Limited and Sanitec Engineers Private Limited, our partner for SFE Process in India. He will bring us for this great webinar over 35 years of leadership in advancing India's process engineering landscape. Under his guidance, both companies has emerged as leading suppliers of ultra-pure water system and customized membrane and chromatography solution for the pharma, biopharma, and nutraceutical industries. He has been instrumental in localizing advanced purification technology through this collaboration with global innovators like Novacep and Aplexion, significantly strengthening Indian capabilities in high-value separation processes. Notably, Mr. Badani pioneered the introduction of batch and continuous prep scale chromatography for APIs, peptides, and functional ingredients at the production scale in India. So thank you everyone for joining us to this session. And to all our audience, this is the first webinar we are organizing on the topic in India. It will give you an introduction to the technology with few examples and feedback of our experts. Here we will remain fairly general, so I strongly encourage you to follow us on LinkedIn to participate in future webinars where we will study more specific cases, going deeply into details, process, and application. As you know, past the last years, all the pharmaceutical industry moves toward more efficient and sustainable processes. SFC stands out as a powerful enabler of green chemistry. By significantly reducing the reliance on organic solvents, SFC offers not just performance gains, but also environmental and economic advantages. After discussing about the current framework and horizon in Indian pharmaceutical ecosystem, we will look more in depth the SFC technology and also highlight some case studies of SFC purification at preparative scale. Be sure to stay with us until the end. We will also have at the end of this webinar a Q&A session where you can ask your question and specific remarks, engage directly with our speaker. Once again, thank you for being here, and let's start to our deep dive into supercritical CO2 and future of chromatography in pharmaceuticals. Thank you.
So, thank you, Jean-Christian, for this introduction, and thank you everybody to be there with us during this webinar. So I'm Jeremy Lagrue, and firstly, I would like to make the focus because before to talk about SFC, supercritical CO2 chromatography, we will focus about CO2. What is the good argument of the CO2? CO2 actually under pressure and after 31 degrees is a very nice solvent. It's mainly used for extraction, sterilization, or also chromatography in this case today. And we have no smell. We have the capacity actually to replace a lot of organic solvent. And this is the main advantage of this technology. It's stable. We have no oxidation, no color, no flavor. So where is from CO2? Why we usually say that it's ecologic? Actually, CO2 is a co-product from waste from industrial plants. So the CO2 that we will use in our system should be already in atmosphere. That's why we say that supercritical CO2 technology it's mainly a green technology. So now supercritical CO2 it's rich after two parameters above 31 degrees for the temperature and 73.8 bar. When we reach this state, supercritical CO2 is not a liquid and not a gas. It's the both combination of the diffusivity and about a very high density make it a very nice solvent. So thanks to this high diffusivity and low viscosity, we usually improve and have a faster chromatography. So regarding SFC, you probably know perfectly how works an HPLC or a HLC. So we need a mobile phase. On supercritical CO2 chromatography, the solvent CO2 is a mobile phase. That will help actually to reduce a very low quantity of organic solvent. And we will save a lot of time during the method of chromatography. It's all very known today for analytics, and SFC process and Sanitec actually work a lot to develop this technology for large-scale production. So we always use Mollier diagram of CO2 to try to explain this technology. This support is very nice. That explain the four states of the CO2, the liquid, the supercritical, the gas, and also the solid. At the left, you have the pressure in logarithmic scale. The blue curve is the isotherm of temperature, and the green curve is the density of CO2. So supercritical CO2 is very nice solvent. Why? Because it's probably the only solvent that we can change the density if we change the parameters of the pressure and the temperature. So all the blue curve, it's a zone where we will be able actually to change property of supercritical CO2. So now, to explain how it works with the PNID. So in the point number 1, we took CO2 from a cylinder or from a tank. It's usually between a mixed liquid and gas. To be able to compress CO2, we will decrease the temperature to reach a liquid state in the point number 2. Then, thanks to the pump, we will increase the pressure and reach the point number 3. After, we will heat the CO2 at the temperature we want, and we will arrive at the input of the column in supercritical phase. As you can see, regarding other SFC or classic SFC, you already know. For the SFC from our company and our partner Sanitec, we have developed a very nice innovation. It's high pressure supercritical fluid chromatography. What is different? Usually, and mainly, everybody use pressure inlet of column between 100 bar and 150 bar. And during the CO2 cross the column, actually, we have a delta P. The point number 5, it's the pressure after a column, so output of the column. So when we use high pressure chromatography, actually, even with the delta P, the CO2 will stay in supercritical phase during all the separation in the silica in the column. But when we use a low pressure, actually the point number 5 will be very below, and the density of the CO2 will be close to the gas. So that's why in classic SFC, usually we need to keep a coeluent with a high quantity of flow compared to the CO2. So thanks to our innovation of high pressure supercritical fluid chromatography, we will be able to continue to decrease the quantity of coeluent need for a separation. So then, after the separation in the column, we will decompress the CO2 at 50 bar, and the CO2 will be evaporate to come back in the gas phase in the separator. In the separator, all the product, the API and the solvent, will be going down by gravity, and the CO2 will stay at the top to be able to coming back in the recycling loops. For our system, recycling CO2 is really important, and it already included in all our system. So for us, it no make sense actually to don't recycle, to have no recycling of CO2. So thanks to have this mobile phase continuously recycled, we will also save operational cost. Regarding the polarity, it's really important, as also the phase on the column. But usually, thanks to our innovation, we will be also able to manage a free solvent method of chromatography. Mainly, as you see, if we use CO2 alone as a mobile phase, we will focus only on lipids, essential oil, and no polar molecules. Definitely, even in SFC, as we need to do purification of polar molecules, we will need to increase the polarity of the mobile phase. And for that, usually everybody will add methanol. But at SFC process, actually, we trust to be able to use also other or solvent as ethanol or water as a co-solvent. Why? Because we know that the methanol also have a bad effect on the silica and on the lifetime. So if we are success thanks to the high pressure of chromatography to use ethanol and water, we will be able to extend the lifetime of the silica. So as you see, there is many lot of advantage on supercritical CO2. We can change with the pressure and the temperature, the density of the CO2. And if we had a coelement as ethanol, water, or methanol or other solvent, we are able to increase the polarity on the mobile phase. So that's made today that SFC is really a large possibility of molecules from no polar to polar molecules of chromatography. So now I will let the power to Mr. Nilesh.
Thank you, Jeremy. Thank you, Joe. Thank you for making me the part of this webinar. Good afternoon, friends. I'm Nilesh Badani. As we all know that Indian pharma industry is one of the important industry globally, and we are supplying more than 20% of global generic medicine to more than 200 countries. And we have a very ambitious plan to reach to 130 billion by 2030. Typically, I would say the Indian generic medicine manufacturers, API, nutraceuticals, they've been using quite a lot of solvents for their either extraction, purification, application. Today, there is also kind of need to control the usage of the solvent. There is a pressure to improve the environmental conditions, or I would say the reducing the organic solvent usage, and also comply with the domestic as well as the international sustainability goal in terms of the emissions, in terms of the pollution, in terms of controlling all the effluent being generated. There are also specific, I would say, guidelines which are released by the Central Pollution, Regulatory, as well as the US FDA and EMA are also, concerned about the effluent which are being generated. Uh, Next slide, please. So there is a, I would say, if we consider, I would say, Indian pharmaceutical industry, the solvents generally which are, consumed are the acetonitrile, methanol, IPA, dichloromethane, hexane, and if you, I would say, consider something like in terms of a product which is being used and the waste which has been created, we need to maintain certain level of the ratio. So today I think it ranges from 25 to 100 kg of waste per kg of the product which is being produced, which is quite, I would say, considerably high. There is also concern for the high VOC, volatile organic compound. There is also, issues with respect to the safety, fire, toxicity, and there is it also, increases the water pollution and the concerns for the occupational hazard. Also, it has been recent studies have shown that in terms of contribution of the solvents in the manufacturing of a product, or I would say the purification, it contributes almost 30 to 35%. Like people sometimes think that, for example, if I take a chromatography, process, people think that chromatography equipments is very expensive, or the resins are very expensive. But sometimes we don't realize that the solvent which are used as an elution, they contribute more than 35% of the cost. And this is also one needs to, limit the use of fantastic technology like chromatography, which everyone would like to have it, but because of the certain cost factor, they are not, I would, able to move towards that. I would say that this also makes us to think like an alternative to the solvents, and if something which we can use it in our process for the separation purification, it can really bring in a lot of value in the manufacturing. And this is where I would say the supercritical chromatography or can play a very good role. And I would like to assure you that, I would like to say that the technology or the certain, I would say, innovation which has been bought by SFE process, that can really help us to improvise our process and the purifications. So I think the greening Indian pharma industry, that has been everywhere. We have been hearing that we the industry has to move towards the green chemistry, green technology, green processes, environmentally friendly processes. And this is a supercritical, carbon dioxide or supercritical CO2 can help us in achieving this. It is a cost efficient, CO2 is cheap, it is an inert, and it can be recycled. So all the, I would say the equipments which are being manufactured, they have a inbuilt feature where the CO2 is recycled. I would say it's a gentle and versatile, ideal for the thermo level, APIs, small molecules, and then the natural products. And it's quite, I would say now people have been using it. People have been developed lot of methods, lot of processes, and it's now people are really in a mood to scale it up to a large scale. Also, in terms of efficiency, it also yield, we get higher yields, and especially I would say efficient for chiral and achiral, I would say, separation. And already as mentioned, very friendly for the pollution control board. It meets the global guidelines and safer for the workers. So I will now ask Jeremy to take us into a kind of a deep dive for further into in terms of the technology aspect as well as with some case studies. Thank you.
Thank you so much, Nilesh. So regarding all this advantage of the SFC we discussed today, you need to know that this technology is known from very long time. And we try to understand why actually it's not already largely developed. Actually, we discovered that there is three issues we discovered on the scale up of this technology. And the first one was some difficulty of reliability and technical issue on scale up for SFC system. That's the first blockage actually for some people. This is what we hear usually after discussion with some people. The second point is SFC is not very known by everybody, and actually that need a support and training from our company to be able to give all the knowledge and be able to be totally autonomous to work with this technology. And the last one is to have a really nice service on support for after sales, because when you invest in a system for production, you expect to have a system running 24 hours with no issue. And if you have an issue, we need to fix it very quick. So firstly, we work on the 3 points to be able to increase the trust on this technology because it's really wonderful and could be help you to save a lot of time and money for your application. So firstly, for technical aspect, we discovered that there is some difficulty to have a reliable and on the injection. Usually it's using a pump to do injection, but as you know, a pump have some difficulty with a check valve, can have a cavitation, etc. So we developed a injection tank, really easy and baby-proof to feed the loops. And like this, actually, we have fixed the issue on the injection. The other point, we heard from the market on the difficulty, technical difficulty, is recycling CO2. When we use a large quantity of coeluent, some people explain us that coeluent was also not very well collected in the separator, and it was recycled with the CO2, and that will move the line base on the chromatography. You need to know that thanks to our experience on 20 years old, actually, we have put all innovation to fix the issue and be totally sure that the CO2 recycling is free of any coeluent or API, etc. This is the 2 main points, but there is a lot of also other innovation that we had to develop this technology. As we explained, firstly, we developed high pressure chromatography, and we developed also a very nice software, user-friendly, and we always still happy to discuss with you if you want to implement something or show something. So as you see on the software, very user-friendly. This method of development was developed at 500 bar. And with the CO2, we have a delta P only about 10 bar on this case. But the high pressure was very a difficulty for us, especially on the semi-prep, because we need to use hardware. The hardware we find on the market actually was not able to work at more than 300 bar, and also have not all the security from the regulation of high pressure vessel from Europe. So it was necessary for us to develop our own hardware, and this hardware is controlled by the notified body and respect the DESP about the pressure. Because without hardware, we will not able to make the method of development and very high pressure. So we are very happy to tell you that we have 2 now new hardware available from 500 bar and other and 1000 bar. So the high pressure will help a lot to reduce the quantity of coeluent, and I will explain you later on the case study. So this is mainly for the issue we saw on the market for the technical point. SFC is a lot of developed today on analytics because we saw at Gelen, Shimadzu, Waters develop some analytical SFC very well. But it's mainly developed for chiral separation or screening, but it's not very developed for the moment in SFC prep. There is some company trust on this technology as Aragen, or in Europe we have also KD Pharma have a very large SFC system for production. And our goal actually is to prove you that we can trust on Sanitec and SFE process to develop your production thanks to our support. On the point number 2, to help you to control and know very well this technology, actually, we have a lab with 3 people full time to help you actually to make a method development. So internally, actually, we have extraction from 100 ml to 25 liter. We have fractionation column to be able also to do concentration. We have semi-prep 30 millimeter column of chromatography 1000 bar. And we have everything to do analyze thanks to our HPLC from Thermo Fisher with the data corona and the GC mass spectrometry. So this training and this method of development and this support is really important for us because this is the key usually of the success of your project. So don't hesitate to contact us to start working together. So as this support is really important, actually, with Mr. Nilesh and the company Sanitec, we expect to develop this support directly in a workshop of Mumbai with the team of Sanitec and with Jean-Christian directly on it. So soon, SFC semi-prep 30 millimeter 1000 bar will arrive during a couple of months. And also 500 ml extraction for study scale. Why it's important to have extraction in chromatography? Because sometimes when we work on chromatography, it's a product from an extraction firstly. And if we do also a study of extraction in CO2, that will help us to find directly and save time to find the good parameters on method of purification on SFC. So now I will talk you about 2 methods of development we are able to discuss with you because we do also a lot of other method of development with some company, but everything is confidential. So we are able today to talk to you only on 2 applications. The first one is a purification of omega-3, so EPA, DHA, and DPA. So this request is usually on SFC because it's a product Apollo. This is a resume of a very long job from our company. Probably we work on this project for close to 2 or 1 year. And this is the conclusion, but we still need to improve and do optimization. What is important to show you on this method of development we success to do in high pressure CO2 chromatography. We success to do separation and concentration of DPA and DHA. Usually, if we do that you will use a huge quantity of solvent. And the key of this technology is to try to use the large sheet of compounds. And omega-3 is a good application for us because we can do that totally with no cost, only with the CO2 So S51 is the first collector, S52 is the second one. S53, as you understood, each one is one collector. So after 26 injection, we do the collection of and we compare the analyze with GC mass spectrometry from the crude oil. So we can see, for example, that it's amazing in CO2 that from 3%, we success to increase the concentration of DPR around close to 70%, only with CO2. So we was very proud when we success to do that. And I'm happy to share you a part of the result today. So if you have some question later on this application, don't hesitate to contact us directly. The second application, very interesting for us, it was on astaxanthin. Astaxanthin is obtained by CO2 extraction of Haematococcus algae. Astaxanthin purification is also really interesting because it exists with 3 isomers of these molecules. So after several months of test, what we saw, I show you the last chromatography in free solvent, but we saw the impact on about the quantity of coeluent. We made the method of development at low density with 25% of ethanol, and we success to get a very nice purification. And when we increase the pressure and the density on CO2, and if you keep the same quantity of coeluent as ethanol, actually we will have less of resolution and we will be not able to do separation. But if you decrease the quantity of coeluent, you come back with a nice resolution. This is what it was very nice that we saw that low density with 25% of ethanol was the same result of purification with very high density and 5% of ethanol. So thanks to the high pressure, we success to save 80% of ethanol as a coeluent. And I will show you a resume about the quantity we save during this method. This is what we showed now. It's a test of a free solvent with no coeluent. It was very nice. And it could be also optimized because the last peak, actually, it's product no interesting. So we can have just there after 7 minutes a flush of ethanol to save time and be able to inject back. So regarding this result, is still on the way regarding concentration, but we do collection with 4 collection for the 3 first peak and the last collection for the last one. Really interesting is at the top. We have the chromatography, mass chromatography of the standard of 1 isomer of astaxanthin. And we saw also the chromatography of the crude. So it was interest for us because I guess that the astaxanthin was on the red color, and on the red color you see on the glass vial, it's very at the right side, so on the last peak. But we discovered that astaxanthin was in the middle, and when we do analyze, we saw that we success actually to have 2 isomers and very close to the quality of the standard we buy to do this comparison. And the last one actually is really no astaxanthin. So we are still working on this application because it's really interesting to see that we are able also to separate isomer directly on CO2 supercritical CO2 chromatography. And hopefully on the next webinar or next discussion, we will be able to give you more input. But it was important for me to try to share you parts of the scale-up and how many we can produce and the quantity of solvent used for it. So, as I told you, low pressure with 23% of solvent, it was a method of development, but high pressure with 5 method of solvent was also a very nice method to do purification of astaxanthin. And the last one, in pure CO2, this one is really the worst case because we can also reduce by 2 the method and increase the productivity. But it was very interesting also to see when we do the scale up and calculate of scale up on the SFC 100 millimeter, that even in SFC with 23% of coeluent as ethanol, to produce 69 grams of astaxanthin, we need to use 600 liters of ethanol. So even we use already SFC, it's very huge quantity of solvent. So I can't imagine the quantity of solvent need to be used when we use only HPLC. So directly with the SFC, we are able to reduce very high quantity of coeluents, and that will save time and that will save also operational cost. And you will have advantage on the market with the product with higher margin. That it's really important to show you that the main advantage of this technology is to reduce the quantity of organic solvent. So I would like to say thank you at everybody. Jean-Christian, I let you start to discuss about our conclusion. Or
Okay.
2 minutes,
So
Nilesh. Okay. Nilesh, I'll let you discuss about the conclusion. Thank you so much.
thank you, Jeremy, for going through in a good detail in terms of the principle of the supercritical, in terms of how the system has been designed. You also took us through case studies, 2 case studies, which was quite, I would say, interesting. I think I would say, I would conclude like this, that the supercritical fluid chromatography, it's a nice process, eco-friendly. It is emerging as a scalable process. It is a cost-efficient and benefit. It's a greener, I would say, purification platform, especially for the pharmaceutical and high-value nutraceuticals. It is really a technology to be considered. You also, showed us that in terms of the productivity, because of the faster cycle time, the yields or productivity is much better. In terms of solvent or the consumption also, it is a very less, or because of the innovation which you have built in, you are able to operate at the high pressure without the coarse solvent or a very minimum quantity of a coarse solvent. I would say that today the supercritical chromatography is not a, I would say, a technology which is only at the lab scale, but it is ready to be applied for the industrial scale production. Also, I would say that, as you mentioned, that we are partnered here to support for the ensuring the customers that we ensure that they get a good after sales support, and I would say the reliability or on the equipment and technology is not, I would say, going down to, or I would say, to make them have a second thought in choosing this technology. So with this, I would say that, friends, I will hand over to Jean-Christian, and then we can have some question answer.
So thank you very much, Nilesh G. Thank you very much, Jeremy, for this very nice presentation. Now, as you said, it's time for Q&A. So let's open the floor of question and then see that Mr. Mathieu Giraud already asked a question. So maybe for Jeremy, can you comment on productivity achieved, kilogram of product purified per 24 hours per kilogram of packing material? Maybe we can take back the two examples. One was already explained, but Jeremy, I'll let you reply on this question, please.
Yes, the productivity is really depend also with the method, and that's why also we have a large range of system from 30 millimeter, 50 millimeter, 100 millimeter. But if we discuss about kilograms, usually we will choose a system SFC 50 that we can pack with 30 grams or 300 grams of silica. And we can also increase this productivity if we increase the size of the diameter of column. But it's hard to have a real reply of this question because it is mainly depend about the method of development.
Thank you, Jeremy, for this answer. So do we have any other question in the chat? Dear audience, please feel free to ask any question to our two experts. And if you are feeling shy to ask a question, you can also directly contact us for discussing about your project or your question directly with some of our teams. So Mathieu Giraud said in the chat, there is no answering to my question. My question is not specific from the scale as it is a ratio kg per kg.
Yeah, thanks for this question, Mathieu. We can discuss about it because I have not the reply now and to know with which product we need to discuss. So I will be happy to discuss with you later and reply you deeply in this detail on this question.
Thank you, Sir Giraud, for also responding on the chat. We have one other question from David. So do you have any other case studies that you can share with us, with the audience via your website or maybe here on this webinar?
On this webinar, not, because as I told you, we work also a lot with other case study, but it's confidential. You need to know that we have a very huge project on the peptide purification with support on the French government that gives you financial help on this project. So in future, we will be able to share you more details about the project on purification of peptide. We have we worked with a project no confidential now is the purification of oryzanol, so from an extract from rice. So this is something also after this webinar, Mr. David, if we want to discuss, I will be very happy to share you some data.
Something also that it's not really relevant for Indian market, but we have a lot of case studies on cannabinoid purification because that was one of the main application of SFC. I don't know if you want to react on this, Jeremy.
Yeah, and on cannabinoid purification, actually, we success to have a method development directly for CBD or THC medicals with no coagulant or with very low quantity of coagulant. So this is already developed, but it's mainly we can find also a lot of bibliography on this technology for cannabinoid. But the high density, high pressure chromatography we have developed will help to save a lot of time and a lot of quantity of reagent.
We have also one other question from Mr. Giraud. So what is the typical CO2 percentage achieved for recovery at industrial scale?
Actually, all the CO2 will be recycling. The loss of CO2 is mainly when we do the collection. So, as I say, if we don't collect, we have no loss of CO2. And the purity of the recycling, it's very higher because it's more than 99.5%. This is something actually we do. Even also with the volatile, because we have a lot of project confidential with volatile project. And we do some tests internally with limonene. Limonene actually is worse than the CO2 to do the recycling, and with success to avoid that the limonene recycling with the CO2 on the loops.
One question from Najendra G. We are working on the purification of polyphenols on preparative chromatography, so the alkaloids is not separated. Maybe Najendra, if you can give more context to your question, it will help us to reply for your inquiries.
So definitely, actually, the team of the lab and the system we have is there also to come and test this high pressure CO2 chromatography with our knowledge. So if we are happy to do some tests, you can visit in France and work with us, or wait and work with Jean-Christian and Sanitec, where the system will be available in Mumbai.
So Najendra G give.
But of course, we have some knowledge with polar molecules as polyphenol or other alkaloid.
So Najendra G gave few details about the question, few extra details. Will the supercritical CO2 has the capability to separate two compounds which are close to each other, like alkaloids and polyphenols? And have you scaled this technology in any industry?
I can't say you yes, I can't say you no. It's confidential, but we will be really happy to work together on this question.
But maybe just to see again the two different examples we've showcased on this webinar, the molecules that we are separating with the technology are really close in terms of formulas, in terms of configuration. So definitely we can exchange, dear Naranja, if you want, and maybe make a test here in Navi Mumbai once the system will be available for testing your specific inquiry. Any maybe last question? We still have like 4 minutes, so please feel free to ask any question. Just wait for 5 seconds. If not, then we will close this webinar.
Just last thing, regarding the support, as our system is really easy to ship, there is some product we are not able to work in our lab. So sometimes actually we are able to discuss about agreement rental to be able to work directly on your site with your agreement to work with some dedicated product.
So we have one last question from Dr. Parin. Could you summarize how SFC process equipment specifically address scalability challenges from analytical SFC to preparative of production scale compared to competitors?
Yeah. So this question is really interesting. The scalability from analytical to prep. Actually, our company is really focused only on the prep chromatography, but we saw that the method of development made on an analytical system, it can't be directly applied on preparative system. Firstly, the analytical system have a very dedicate high delta P, because the design of all analytical system is very with a small pipe. So even the column made already delta P, all the line on the system made and had also delta P. And when we do the scale up on the prep, actually with our system, we have no delta P with the line. So only the delta P is from the column, and that may sometimes huge difference. So that's why actually we have developed this semi-prep system, so that we can put 20 millimeter or 30 millimeter column, is to be able to work and adjust the method development made on analytical to be able to have a very nice and representative production on the prep system. So mainly during this webinar, I explained that what is different or compare our competitor is we are a bigger company. We have a very huge after-sales service. Is for point one. We are able to support and training you about this technology and help you on method and development. And we developed. We had a lot of innovation from our experience on other application with the injection, with the recycling of the CO2, and we developed altogether the possibility to work at high pressure supercritical CO2, and not only 100 or 200 bar.
Great. So there is no other question. So I think, ladies and gentlemen, we can come to the end of today's session. So I would like to, of course, thank you, our speakers, and also to warmly thank you, our audience. Thank you for your attention. Thank you for your question and your engagement. Of course, please follow us on LinkedIn for the updates, and also for our next webinars where we can showcase some new application, and I hope so for peptide purification. And also to follow our different representation for various events across the world or conferences. Also for our Indian audience, tomorrow is starting Ganesh Stratuti Festival. So from all our team, from Sanitec and from SFE Process, we are wishing you in advance a great 2025 Gambati Festival, and have a nice end of day. Thank you, everyone.
Yeah. Thanks also Frédéric Guichard, because we have also partnerships with the Dietrich to work with very large scale on this technology. Thank you so much.
Thank you. Have a nice end of
Thank you,
afternoon.
everyone. Bye.